Hello everyone and welcome to today's session. And today we'll be discussing the theory related to the chapter Cell the Unit of Life. Okay. So in this session we'll be discussing the NCERT section 8.5.3 as well as 8.5.3.1. Okay. We'll also discuss the section 8.5.3.4. And this is the part 4 of the lecture. Before starting this lecture, I just want to give you one information that the images section of the materials used in the presentations are for education purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathee@itk.ac.in and we shall remove the content immediately. Please note that parts of the content may have been generated using generative AI, which is experimental. So what are the objectives of today's session? So today we will be talking about the structure and functions of endoplasmic reticulum. We'll talk about rough endoplasmic reticulum as smooth endoplasmic reticulum. We'll also study the role of Golgi apparatus in protein modification and packaging as well. Okay, so these organelles are very important. We'll also understand lysosome and what are the functions of lysosomes in cellular digestion. We will identify the vacuole roles in storage. So these vacuoles, they store some materials. We will talk about that. And what is the role of vacuole in maintenance of the cell pressure? Okay, so these are the objectives of today's session. Okay, we'll be talking about the endomembrane system today. Okay, within this topic, we'll be talking about the endoplasmic reticulum, Golgi apparatus, lysosomes as well, vacuoles. Then we will summarize this entire lecture. We'll discuss some NCERT-based questions related to these topics. There'll be some keynotes and interesting facts for you. And in the end, there'll be some interesting questions. Okay, so let's start the introduction to the endomembrane system. So eukaryotes, they consist of membrane-bound organelles. So membranous organelles have distinct structure and functions. So each organelles have its own function and structure. Some of the organelles forms an endomembrane system due to their coordinated functions. So their functions are interconnected. They are coordinating among themselves. So that's why they are forming a system. So when a system is formed when the components of the system, they coordinate with each other. So organelles in the endomembrane system, they are coordinating with each other to form this endomembrane system. Now, what are the organelles which forms this endomembrane system? So endomembrane system includes endoplasmic reticulum, okay, Golgi complex, lysosomes, vacuoles. Okay, so you can see here, this is the image showing you endomembrane system. You can see the organelles which make the endomembrane system. Which organelles you can see here? See, you can see Golgi complex here. You can see lysosomes here. Okay, you can see the endoplasmic reticulum. See, you can see the smooth endoplasmic reticulum here. This is the rough endoplasmic reticulum. Clear? All right, so organelles which are not the part of the endomembrane system. First is mitochondria. Mitochondria, it is a semi-autonomous organelle. We'll talk about mitochondria also. It is not the part of endomembrane system. Chloroplast also, this is not the part of the endomembrane system. Chloroplast is also semi-autonomous organelle. Then peroxisomes are also not the part of endomembrane system because their functions are not coordinated with the endomembrane system. So these three organelles, they are not the part of endomembrane system because the functions which these organelles perform, they are not coordinated with the endomembrane system. So they perform some other functions. Okay. Now, what are the characteristics of the endomembrane system? So the main feature that defines the endomembrane system is the coordinated functioning of its component organelles. These components or these organelles, they work together in processes like protein synthesis. Okay. Then modification of the molecules, sorting of the molecules, and then transport of the molecules. So these are the major processes that are carried out by the endomembrane system. Clear? So we'll see which organelles perform protein synthesis, which organelles is involved in modification, which is involved in sorting and transport. Clear? So this is, you can see this is like a factory. The molecules are being synthesized and they are being modified. They are being sorted. Okay, sorting is also very important. And then transport. Now, what is the basic structure of the endoplasmic reticulum? Okay, abbreviated as ER. So it is a network of tubular structures which is present in the cytoplasm. Now, endoplasmic reticulum creates two compartments. Okay, so what are these two compartments? First is the luminal, which is inside the ER, and another one is the extraluminar, which is in the cytoplasm. Now this ER, this is a continuous membrane system. You can see this is an image showing you endoplasmic reticulum. Okay, so here you can see cisternae are present. Okay, ribosomes, if they are present, then it becomes rough endoplasmic reticulum. Ribosomes are involved in protein synthesis. Okay, so you can see this is the rough endoplasmic reticulum. The above image is a rough endoplasmic reticulum. Then below this is the smooth endoplasmic reticulum in which ribosomes are not present. Okay, so here cisternae are present in the endoplasmic reticulum. Now, types of endoplasmic reticulum, there are two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. So, rough endoplasmic reticulum has ribosomes attached to their outer surface. Now, RER is connected to the nuclear membrane. So, here the question comes, okay, in your exam, which is the organelle which is connected to the nuclear membrane? So, it is the endoplasmic reticulum. It is connected to the nuclear membrane. What is the main function of rough endoplasmic reticulum? So as this is associated with ribosomes, the main function is protein synthesis and secretion. Prominent in cells that produce a lot of proteins, like pancreatic cells. So where are rough endoplasmic reticulum present? So they are present in those cells in which a lot of protein synthesis is being done. Example is cells of the pancreas. Pancreas also secrete a hormone. Which hormone? Pancreas secrete a hormone which is insulin, which is a protein hormone. Now, next, this image we have discussed, this is the endoplasmic reticulum. Now, next is smooth endoplasmic reticulum. So, it doesn't consist of the ribosomes. Now there are no ribosomes. So, what will be the function of smooth endoplasmic reticulum? So, function is lipid synthesis. Okay, this is the main function. They perform other functions also, but here we are talking about the main function. Now it produces steroidal hormones in the animal cells. Okay, so again a question can come, which organelle synthesizes steroidal hormones? So answer will be smooth endoplasmic reticulum. Now, more abundant in cells involved in lipid metabolism. So obviously, the cells which are involved in lipid metabolism, they will consist of more number of smooth endoplasmic reticulum. Now next, what are the key functions of endoplasmic reticulum? It is involved in compartmentalization of the cells. Clear? So, as we have seen, this ER divides the cell into compartments. Then, functions are protein synthesis and transport done by the rough endoplasmic reticulum. Lipid production is there by smooth endoplasmic reticulum. Cellular organization. It is connected with the nuclear membrane. This is a feature of endoplasmic reticulum. Then, what is the significance here? It is essential for cellular organization. Okay, critical for protein and lipid synthesis. Important for hormone production. See, the smooth endoplasmic reticulum, this was synthesizing steroidal hormones in the animal cells. Clear? So, hormones can be made up of steroids. They can be made up of proteins also, like insulin is a protein hormone. Testosterone, estrogen, these are all steroid hormones. It is vital for cell membrane maintenance and growth. This is very important point, because endoplasmic reticulum is synthesizing lipids also. The cell membrane, also known as the plasma membrane, this is also made up of lipids and proteins as well. Okay, this is an image showing you endoplasmic reticulum. You can see here ER is attached to the nuclear membrane. Okay, this is the nucleus. Nucleus consists of nuclear pores. Here you can see this is the rough endoplasmic reticulum, and this RER is associated with the ribosomes. Okay, these are the ribosomes. Then this is the smooth endoplasmic reticulum. Both have different function. RER protein synthesis, smooth endoplasmic reticulum lipid synthesis. Now let's talk about the Golgi apparatus. Structure. What is the structure of Golgi apparatus? Sometimes called as Golgi complex also. So, this was first observed by Camillo Golgi in 1898. That's why the name Golgi. Now, Golgi apparatus consist of stacks. Okay. So, they consist of flat disc-shaped stacks known as cisternae. Each cisternae is 0.5 to 1 micrometer in diameter. So, micro is 10 raised to the minus 6. Okay, so here you can see this is Golgi apparatus. Golgi apparatus has a cis phase and a trans phase. Remember this. Clear? So, this is the lumen here. This is the cisternae you can see here. Clear? This is the incoming transport vesicle. And these are the secretory vesicles made by the Golgi apparatus. Okay, we'll talk about this image. Now, we were talking about the structure. It has two distinct phases, cis and the trans phase. So, cis is basically the forming phase. Okay, it is this phase is closer to the nucleus. Clear? Now, trans phase is the maturing phase. Okay, and it is the concave side. Then both phases are different, but they are connected to each other. So you can see in this image, one is the cis phase, another one is the trans phase. Okay. Then next, what is the location and association of the Golgi apparatus? It is located near the nucleus. Endoplasmic reticulum was connected to the nuclear membrane but is located near the nucleus. It maintains close association with the endoplasmic reticulum. Okay, because they have to coordinate with each other. That's why Golgi apparatus maintains a close association with the endoplasmic reticulum. Golgi apparatus works in coordination with endoplasmic reticulum for protein processing. So proteins are synthesized by a rough endoplasmic reticulum. Then what happens then? Then the Golgi apparatus helps in processing. Clear? So this image we have already discussed. This is showing you endomembranous system. Here you can see Golgi complex, lysosomes, as well as the endoplasmic reticulum. Now, what are the function of Golgi apparatus? Packaging center. So packaging of materials is occurring here. Now, it receives vesicles from endoplasmic reticulum at the cis phase. Okay. So we have discussed the Golgi apparatus had a cis phase and a trans phase. So the cis phase will receive the vesicles coming from the endoplasmic reticulum. Now, Golgi apparatus processes and packages the material. Now, then it delivers to either intracellular destinations or outside the cell as well, means extracellular. Clear? So it is involved in processing, packaging and transport as well. This is the function of Golgi apparatus. Okay. So what are the functions of Golgi apparatus? Okay. Second is protein modification. It modifies proteins received from endoplasmic reticulum. So endoplasmic reticulum is synthesizing the proteins, and the protein modification is also done by the Golgi apparatus. Now processing occurs as materials move from cis to the trans phase. So when the materials, okay, the proteins is moving through the Golgi apparatus from cis to the trans phase, they are modified. Now it is also involved in complex molecular formation. So it is a key site for producing glycoproteins and glycolipids as well. So these are complex molecules. So glycoproteins consist of a carbohydrate molecule also. Carbohydrate molecule is attached to proteins. Then glycolipids is when the carbohydrate molecule is attached to the lipids. These are complex molecules. So Golgi apparatus is also a key site for producing glycoproteins and glycolipids as well. It is essential for final protein processing before the cellular distribution. Before these molecules are distributed to intracellular destination or to extracellular destinations, these proteins are processed inside the Golgi apparatus. Here you can see this image showing you Golgi apparatus. You can see the cistern here. Now let's talk about the lysosomes. So lysosomes are membrane-bound vesicular structures. Okay, these are kind of vesicles formed by the process of packaging in the Golgi apparatus. Clear? So this is what is lysosomes. Now the isolated lysosome vesicles are very rich in almost all type of hydrolytic enzymes. So these lysosomes, which are basically vesicles, which are bounded by a membrane, so they are membrane bound. And these lysosome vesicles, they are known as suicidal bags. Okay, why? Because they consist of almost all kinds of hydrolytic enzymes, such as hydrolases, okay, like lipases, like proteases, like carbohydrases. So lipases, what is the function of lipases? Lipases will degrade the lipids. Proteases will degrade the proteins, and then carbohydrates, they will degrade the carbohydrates. Clear? Now these enzymes are optimally active at acidic pH. Okay, so every enzymes are basically, most of them are proteins. Okay, so these enzymes, they function at a optimum pH and optimum temperature. So all these hydrolytic enzymes, they work at acidic pH. Clear? Now next, now function of these enzymes is to digest carbohydrates, proteins, lipids, nucleic acids also. Okay, so there are enzymes known as nucleases which can degrade nucleic acids also. Now what are nucleic acids? So examples of nucleic acid will be DNA and RNA. These are nucleic acids. DNA is a genetic material. DNA is made up of nucleotides. So it is a polynucleotide which are joined together by phosphodiester bonds. Okay. Now the worn out cell parts are also digested by these enzymes. Okay, these are sometimes referred to as suicidal bags of these cells, the cells which are damaged. Okay, or the worn out cell parts, these are digested by these lysosomes. Okay, when the lysosome bursts, what will happen if the lysosome will burst? Or if the enzymes, they come out of the lysosomes, then what will happen? Okay, will the cell survive or will the cell degrade? Cell lysis will occur. So what will happen? So you can see this lysosome consists of enzymes which are activated at the acidic pH. Clear? So whenever they come out of the lysosomes into the cytoplasm, they will cause the degradation of the cell. So the worn out cell or the damaged cell, they are digested by these enzymes. Though these lysosomes are also known as the suicidal bags of the cell. Here you can see, this is an image showing you lysosomes. Clear? So these lysosomes, these are like vesicles, you can see. They are membrane bound. Okay. These are the hydrolytic enzymes present in the lysosomes. Clear? They are also known as suicidal bags. So question can come in the exam, which of the following is known as the suicidal bags of the cell? So answer will be lysosomes. Now let's talk about the vacuoles and what are the functions of vacuoles. So vacuoles are membrane bound space in the cytoplasm. So they are also membrane bound. Now what is the membrane of vacuole known as? Okay, the membrane of the vacuole is known as tonoplast. Okay. Now this is enclosed by a single membrane known as tonoplast. So tonoplast helps in the you can say entry of the molecules or the ions into the vacuole. Now these vacuoles, what are the function of these vacuoles? They will store water. Okay, they can store some sap. They can store waste products also. And vacuoles, they are of different types also. Clear? So all the waste products, they can be stored in the vacuoles. These waste products can damage the cell. So due to metabolic activities or due to the biochemical reaction, lot of waste products are being produced. If these waste product, they you can say they are present in the cytoplasm, then they can damage the other organelles as well. So what does cell do? They store these waste products or waste materials into the vacuoles so that they can protect the cell. Okay, because these waste products can be toxic. In plant cell, the vacuoles can take up to 90% of the cell volume. So you must have studied about the plant cell and the animal cell. One of the difference was that the plant cell consists of a large central vacuole. So most of the space of the plant cell, this is occupied by the vacuoles. So vacuoles can take up to 90% of the cell volume. But in the case of animal cell, the vacuoles, very small vacuoles are present actually. Now, tonoplast. What is the function of tonoplast? Tonoplast move ions against the concentration gradient. So, whenever the molecules are being transferred against the concentration gradient, so then the energy is utilized. So, it will be a active process. So, tonoplast, what is the function of tonoplast? It will move ions into the vacuoles against the concentration gradient, means utilizing energy. It will utilize energy, and then it will transfer the ions against the concentration gradient. Now, higher concentration of materials inside than in the cytoplasm. So you can see why the ions are being transferred, why the ions are being moved against the concentration gradient, because inside the vacuoles the concentration of the materials is more as compared to the cytoplasm. So energy is required to transfer the you can see this waste product against the concentration gradient, because the cell, it is forcing these molecules to enter into the vacuoles. Here you can see this is the image showing you vacuole. Okay, so this is the cell, this is the central vacuole. Oh, okay, this is the vacuole, and you can see this is the membrane of the vacuole, which is known as the tonoplast. Okay. Now, let's talk about the vacuoles in other organisms. So, amoeba consists of contractile vacuoles. So, vacuoles can be of different type. First is contractile vacuoles in amoeba. So, amoeba is a eukaryote. Okay, it belongs to kingdom Protista. So, central vacuoles are present in amoeba. And what is the function of central contractile vacuole? It is important for the osmoregulation and the excretion. Clear? So, you have to remember the function of this contractile vacuole. By osmoregulation, this amoeba can maintain homeostasis. Now, next is food vacuoles. They are formed in many protists by engulfing the food particles. Okay, so this food vacuole will store the food materials. So whenever they engulf the food, they are stored, these food vacuoles are formed. This food is stored in these food vacuoles and then they are digested. Now, so you can see this is amoeba. This has pseudopodia. You can see these have pseudopodia, which are false feet. So with the help of the pseudopodia, the amoeba can move also, and it can engulf the food also. Okay, so here you can see this is a food particle. The food particle is being engulfed by the bacteria with the help of pseudopodia, and then this food particle is stored in the food vacuoles. Here this is the contractile vacuole present in amoeba. So amoeba, as I have told you, this is a protist. Protists are what? Protists are unicellular eukaryote. So this is a eukaryote. That's why here a nucleus is shown. Clear? Eukaryotes, they have a true nucleus within which the genetic material is present. So they have food vacuoles also, they have contractile vacuoles also, by which they do osmoregulation. Okay, this is clear. Okay, so this was about the endomembrane system. We have discussed the role of the organelles. Okay, we discussed about the endoplasmic reticulum, Golgi apparatus, vacuoles, as well as the lysosomes. Now let's summarize the entire lecture. So first, endoplasmic reticulum, it is a network of membranous tubules. Okay, ER can be of two types, rough endoplasmic reticulum, a smooth endoplasmic reticulum. Rough endoplasmic reticulum will have ribosomes, and the function is to make proteins. Next is smooth endoplasmic reticulum. It doesn't consist of ribosomes, so the function is to make lipids. Now, endoplasmic reticulum transport material within the cell Golgi apparatus. What are the main function? Processing and packaging of the proteins, which are synthesized by the rough endoplasmic reticulum. So this Golgi apparatus receives the vesicles coming from the endoplasmic reticulum, and then when these, you can say, these proteins are modified inside the Golgi apparatus. Okay, Golgi apparatus is also involved in the formation of complex molecules like glycoproteins and glycolipids. Clear? Now, modified proteins and lipids. So these vesicles enter through the cis phase and they come out through the trans phase. And in between, they are modified within the Golgi apparatus. Then lysosomes, they contain the digestive enzymes. They can break down the cellular waste because they consist of these digestive enzymes. That's why they are known as the suicidal bags as well. They aid in cell recycling. Vacuoles, they store, they are the storage compartments. Okay, they maintain the cell turgor also by storing water. Clear? Now, they store water, nutrients, and waste materials as well. Vacuoles are large in plants, and only small vacuoles are present in the animal cell. Now, let's discuss some NCERT-based question. So first question is, which statement about the endomembrane system is false? Means which statement is incorrect. So what is the first option? Endomembrane system helps in cellular secretion. Next is it helps in protein synthesis. Option number C, generates cellular energy. And option number D, it helps in intracellular transport. What would be the correct answer? Okay, so let's have a look at the answer. Option number C is the correct answer. It generates cellular energy. Okay, let's have a look at the explanation. So, cellular energy generation is primarily the function of mitochondria. So, this is false statement. ATP is generated by mitochondria. Mitochondria is known as the powerhouse of the cell. So, endomembrane system doesn't perform the function of generating cellular energy. Okay, mitochondria are not the part of endomembrane system also. Endomembrane system includes which organelle? Endoplasmic reticulum, Golgi apparatus, lysosomes, and vacuoles or vesicles. Clear? Nuclear membrane, nuclear envelope as well, cell membrane as well, in addition to the other organelles. Okay, so the components of the endomembrane system, they are involved in protein synthesis, secretion, transport of the proteins, but not energy generation. Next, the suicidal bags of the cells are, this is a very straightforward question. Options are mitochondria, chloroplasts, lysosomes, and vacuoles. What will be the correct answer? So let's have a look at the answer. So option number C will be the correct answer, lysosomes. So lysosomes are suicidal bags because they contain digestive enzymes that can break down these cellular components. So they consist of proteases, They consist of nucleases, carbohydrases, lipases. Now, if lysosome ruptures, then what will happen? These enzymes are released and can destroy the cell from within, leading to cell death. This is also known as autolysis. So, when the cells are damaged, then these enzymes are released and this will degrade the cell. This process can either be accidental, okay, or it can be programmed also. Okay. Now, the term lysosome was coined by Christian D. Dewey, who discovered lysosome in 1955. The nickname suicidal bags reflects their potential for cellular self-destruction when the cells are compromised. Next question, the space inside the ER is called? Options: matrix, lumen, cristae, or stroma. What will be the correct answer? So let's have a look at the answer. Okay, answer is option number B, lumen. Now, the lumen is where proteins and other molecules are processed and transported. The hollow space inside the endoplasmic reticulum is called the lumen. This is the correct answer. The other options are incorrect because matrix is found in mitochondria. Cristae are inner folds of mitochondria. Stroma is found in the chloroplast. Next question, the membrane-bound organelles where protein synthesis occurs is. Okay, they are talking about an organelle where protein synthesis is occurring. So options are smooth endoplasmic reticulum, rough endoplasmic reticulum, Golgi apparatus, or lysosomes. What will be the correct answer? Alright, so let's have a look at the answer. Answer is rough endoplasmic reticulum. Option B is the correct answer. So rough endoplasmic reticulum has ribosome attached to their surface, and ribosomes are the site of protein synthesis. Okay, ribosome attached to the mRNA and they translate the mRNA into proteins. ARIA specializes in protein synthesis and processing. The rough appearance is due to the presence of ribosomes. Now, proteins which are made here are exported or used in the cell membranes. Next question, cell secretion involves which sequence of organelles? So, these are the four options given to you. What will be the correct answer? First is ER to Golgi, then vesicles, then Golgi, ER, and vesicles. Option number 3, vesicles, ER, and Golgi. Fourth is ER, vesicles, and Golgi. What will be the correct answer? So let's have a look at the answer. Okay, answer is option number A. That is correct. ER to Golgi to vesicles. So proteins are synthesized in the rough endoplasmic reticulum. Then proteins move to Golgi apparatus for modification. And then modified proteins are packaged into secretory vesicles. These vesicles then transport proteins to the cell membrane. Okay, proteins are released from cell via exocytosis. Okay, this is a standard secretory pathway in the eukaryotic cell. Like human cell is also eukaryotic cell. Now let's talk about some keynotes. So endoplasmic reticulum membrane makes up over half of the cellular membranes. Clear? So this is very interesting facts. Now rough endoplasmic reticulum can detach ribosomes. and then they can transform into smooth endoplasmic reticulum whenever required. Golgi apparatus reforms and flattens about every 20 minutes. Now, plant cell, they have larger vacuoles which occupy 90% of the cell volume. Lysosome contain over 60 different type of hydrolytic enzymes. The endoplasmic reticulum can directly connect to nuclear and membrane through nuclear pores. Now, vacuoles can fuse together to form large compartments. Okay. Now, Golgi orientation determines cell polarity in many cell types. Okay. Now, there are some interesting questions for you that will enhance your critical thinking ability. So, you should think about these questions. First question is, what happens to cellular waste if lysosomes stop functioning? What will happen to the cell? Will it survive or will it die? Next, why do plant cells have larger vacuoles than animal cell? What is the reason for that? Why plants are having larger vacuoles? Now, third question is, how do proteins maintain their correct folding while moving through the endoplasmic reticulum? And the next one is, what's the relationship between ER stress and disease? So, is there any relationship between endoplasmic reticulum stress and some diseases? Next is, how do vesicles know their correct destination in this cell? Means, how do they know where to go? Okay, like vesicles, they are released from the ER and then they go to the cis phase of the Golgi apparatus. So, how is this happening? How is this coordination occurring? Okay, how do they know where to go? Clear? So, think about these questions. So, now we have discussed the theory. Let's discuss some DPP related to these topics. So, let's begin with the first question here. So, first question is, which of the following organelles is not the part of the endomembrane system and why? So, what would be the correct answer here? These are the four options given to you. Golgi apparatus, it is not involved in lipid synthesis. This is the reason. Second is mitochondria, its functions are not coordinated with ER and Golgi. Third is lysosomes, they do not interact with the endoplasmic reticulum. And fourth is vacuole, they are only present in plant cell. What will be the correct answer? Which organelle is not the part of endomembrane system? Okay, correct answer will be option number B, mitochondria. Mitochondria is not the part of endomembrane system because mitochondria is known as the powerhouse of the cell. It generates energy in the form of ATP. Then next, rough endoplasmic reticulum is primarily involved in lipid and steroid hormone synthesis, detoxification of drugs and poison, protein synthesis and secretion, and the last one is storage of calcium ions. What would be the correct answer? See, option number C will be the correct answer. They are involved in protein synthesis and secretion because rough endoplasmic reticulum is associated with ribosomes. Ribosomes are involved in protein synthesis. Next one, the Golgi apparatus is structurally and functionally closely associated with endoplasmic reticulum because it synthesizes proteins that are transferred to the endoplasmic reticulum. Now, vesicles from the ER fuse with the cis phase of the Golgi for material processing. Third one is Golgi provide energy to the ER for lipid synthesis. Fourth one is the trans phase of Golgi produces ribosomes for the ER. What will be the correct answer? Alright, so you can see here, among these four options, option number A and B seems right. Okay. Other two options are not correct. So answer will be from option number A and B. What would be the correct answer? See, endoplasmic reticulum is secreting proteins. Rough endoplasmic reticulum secretes proteins. And then these proteins, they are processed in Golgi apparatus. So keeping this in mind, you have to answer this question. Okay, so answer will be option number B. Vesicles from ER fuse with the cis phase of the Golgi for material processing. Next one, lysosome contain hydrolytic enzymes that are optimally active at the acidic pH. What will happen if these enzymes break or if these enzymes are leaked into the cytoplasm? So option number D, they would become inactive due to the neutral pH of the cytoplasm because they function at the acidic pH. Next is they would digest cellular components during autolysis. Third one is they would fuse with the mitochondria to produce ATP. And fourth is they would be transported back to the Golgi apparatus. What will be the correct answer? Okay, the correct answer here will be option number B. They will digest the cellular components, causing autolysis. So that's why they are known as the societal bags. Okay. Now next one, in plant cell, vacuoles occupy 90% of the cell volume. The tonoplast membrane is critical for. They're asking the function of the tonoplast membrane. First in, they store hydrolytic enzymes similar to lysosomes. Second is, they actively transport the ions into the vacuoles against the concentration gradient. Because the vacuoles, they already have high concentration of the material. Next is option number C, synthesizing glycoproteins and glycolipids. And last one is packaging proteins for secretion. What will be the correct answer? They're asking the role of tonoplast here. Here the correct answer will be option number B. Tonoplast involved in the active transport of the materials or ions into the vacuoles against the concentration gradient. Means this is an active process. Energy is being required here. Alright. So with this, we have discussed the DPP also. Okay. So we'll end this session here. I hope this session was quite knowledgeable for you. We'll meet you in the next session. Thank you everyone for attending this lecture. Thank you very much.